The process of transcription is very simple. First, the DNA must be split. This is done by helicase, represented by the green spherical shape. This breaks the hydrogen bonds between the two nucleotide bases, otherwise known as the unzipping. Only one of the two nucleotide strands are used in the synthesis of mRNA, otherwise known as the unzipping strand. The transcribing strand, also known as the template strand, and the non-transcribing strand is known as the coding strand, because its sequence of nucleotides looks like the protein codon sequence. Polymerase, represented by its black shape, attaches to the DNA at the promoter site. Polymerase moves along the cistron in the 5'3" direction. It passes over the nucleotides in the template and builds up mRNA by adding complementary nucleotides, i.e. adenine to uracil and cytosine to guanine, creating mRNA strands. As the polymerase moves down, the hydrogen bonds behind it, between the two nucleotides, reform. The polymerase continues until it reaches a special stop sequence called a terminator. At this point, the enzyme detaches and the mRNA peels away from the DNA. The process of translation is a complex one, which happens in the ribosome, located inside the cytoplasm. Each ribosome, represented by the blue color, has two subunits, which are a small binding site for mRNA and a large binding site for tRNA. mRNA, shown by the yellow strand, carries the information in the form of codons, represented by the green lines, which specifically correspond to the amino acids used. A ribosome then moves along a strand of mRNA in the direction of 5'3", reading the codons one after another. tRNA, depicted by the red shapes, takes the amino acids and brings them to the ribosome, and these are then added onto the growing polypeptide chain. The ribosome allows two molecules of tRNA to combine with the mRNA strand at one time, i.e. two red shapes both latching onto the yellow mRNA strand. One is holding the chain in the correct spot, and the other is carrying the amino acid to the polypeptide chain. A peptide bond is formed between the attached amino acids, shown by the brown squares, and then the mRNA moves sequentially through the ribosome, releasing the tRNA.